Connects talk. I'm Paul Weiss, coming to you from UCLA in Los Angeles, California. And we're continuing our month theme of applications of nanotechnology. And today, I'm delighted to introduce UCLA alumnus and nanomedicine star, Professor Justin Haynes. Professor Haynes is the Lewis J. Ork Professor and Director of the Center for Nanomedicine at the Johns Hopkins University School of Medicine. He holds faculty appointments in the departments of ophthalmology, biomedical engineering, chemical and biomolecular engineering, neurosurgery, oncology, and pharmacology and molecular sciences. He holds more than 150 patents on technologies to make drugs and gene therapies safer. Several of these have been approved by the US Food and Drug Administration and have already helped many patients. He's a fellow of the National Academy of Inventors, the Controlled Release Society, which he also served as president, American Association of Pharmaceutical Scientists, International Academy for Medical and Biological Engineering, and the American Institute of Medical and Biological Engineering. He was named one of the world's top 100 young innovators and leaders in technology and business by Technology Review, and an Edward C. Nagy investigator by NIBID. He's a friend, and what I hope also comes through today, in addition to being a creative scholar, he's also such a kind and thoughtful person. So let me now turn the floor over to Professor Justin Haynes. Welcome.
Thanks so much, Paul. I appreciate it. Let me try to share my screen here. Okay.
Perfect.
All right, great. Hello, everybody. This is, I want to thank Paul and also Alexang for inviting me here today to tell you some of the work that we're doing. Quite an opportunity to talk to so many of you, and really looking forward to the panel afterwards as well. So I'm going to tell you a little bit of some different stories that have happened to me over my career today. And I want to share them with you because it has a theme. And when I look back over my career, I realized the things that I had done that had been the most impactful and most helpful maybe to people was when, I and my colleagues looked at problems differently. So problems that had, in some cases, very strong dogma that would suggest that, a certain approach was the best approach to create a new, for example, drug delivery system for a disease. And it didn't always make perfect sense to us in certain cases. And so we, went deeper, learned about the biology, talked to clinicians, worked with other various scientists from different backgrounds, and oftentimes ended up challenging those beliefs and found something new. And, I would say leads to innovation. And so that's kind of the story I want to tell, and I'm going to try to do it through a few different examples in my career. So before I get started, I just want to introduce the Center for Nanomedicine at Johns Hopkins. I started the center a little bit more than 10 years ago at the School of Medicine, and it's grown, and it's a fantastic place to come and work. For any of you interested, we are on the heart of the medical campus, but we have a ton of engineers and scientists in our building with us. We're trying to train the next generation of leaders that will advance science and human health. As part of this, we've done a lot of good basic science for sure, but we've also had a mission of trying to do science that can be translated to help people. And one of the things that's resulted in is actually many companies being launched based on different technologies that my colleagues and I have developed over the years. And we've received money from sources like the NIH, but it's nothing compared to the amount of money that gets invested into these companies to make products that can actually help people. So that I'm happy to say that there are clinical trials that have been occurred or ongoing for more than 10 different diseases based on work that we've done, and hopefully that list will keep expanding. I have some financial interests to disclose. Hopkins requires that I disclose that I have, own stock in these companies, and in many cases, they have some sort of advisory role, or even in certain cases, more active roles. And so I own stock, and that my conflict of interest is managed by the Johns Hopkins Conflict of Interest Committee. Okay, so to dive into it, I need to define a couple of things first. One is I'm going to be saying unmet need a few times here. And so what I mean by that today is an unmet need is a need for an improved method of treatment of a disease, for example, of a medical disease. And I'm also going to say then what a drug delivery myth, and I'm going to be a little provocative with these, but I'll say that a drug delivery myth is an incorrect dogma that can prevent innovation. So unmet need number one goes back to my days when I was a graduate student at MIT, working with Bob Langer and Dave Edwards. And there was a real need at that time for inhaled medicines that could provide a more sustained or controlled, what we call controlled release of drugs that could either treat the lungs themselves for a variety of diseases, or that could go into the blood from the lungs, much like when we breathe in air, the oxygen gets into the blood from our lungs. We'd also like to deliver drugs to the blood from something you could inhale. But at that time, there was really no way to get sustained release, for example, into the blood. So that was the myth, that controlled release via the lungs was not possible. And why would we even want to do this? Why was there so much focus on inhalation? Some people call it the other oral route because unlike air, when you're usually breathing it through your nose, when you inhale medicine, you usually breathe it in through your mouth. So it's another way to get drugs into your body through something that you put into your mouth, actually. Advantages of the delivery to the lung are the surface area of the airways in the lungs is enormous. So the surface area is about 140 square meters, and that's about the size, the area of a tennis court. So we have a huge amount of surface area for absorption of drugs in the airways. There's a very mild pH, unlike the stomach, and reduced enzymatic activity in the lungs. So that can favor drugs being delivered this way and remaining intact. The pulmonary mucosa is also permeable even to proteins, and quite large proteins. This is mainly in the alveolar sacs, which are the very ends of the lungs where you have oxygen exchange. The transport distance is very short, less than 2 microns there, such that even proteins can get into the blood when you inhale them. And another advantage that is partly because of these various things that I've just mentioned is that you can get high bioavailability. It means a lot of the drug that you inhale can actually get into your systemic circulation and become available to the body relative to oral delivery. Limitations, though, are inefficient delivery to the deep lung, especially if you make it into a particle for drug delivery. And particles that reach the deep lungs are typically rapidly destroyed. And this is why people thought you couldn't really get long-term delivery. So why is it so difficult to deliver particles? And so first of all, we have to know that the dogma was very strong at the time on particle size. And it was thought that if the particle was greater than about 3 microns, when it was inhaled and it came into the trachea here, goes into the bronchi, and then goes and branches into all these different branches into the lungs until it gets to the ends of the air sacs. If the particles were larger than 3 microns, they were thought to just come out of the airways, and I'll show you why in a second, in the upper airways. They impacted very quickly because of their large size and mass. If they're 1 to 3 microns, they could make it oftentimes into the deep lungs, assuming they could be broken up into those single particles from the inhaler, then they could make it into the deep lung. But if they're too small, like less than 1 micron, they could go and navigate all these different twists and turns through the lung airways and get to the alveolar sacs, but then they would be too light to actually fall by gravity, which is the last step for a particle to deposit in the right size, to fall by gravity and therefore impact before you just exhale it. So if it was too small, you would inhale it all the way and then it would stay suspended in these sacs and then you just exhale it out. So it was really thought you had to have 1 to 3 microns. Well, a problem with that is that any particles that were greater than 3 microns would get stuck, in the airways before they reached the sacs. They would land in mucus, and mucus would just clear them out of the lungs very rapidly, on the order of hours. If the particle was small enough and some got into the alveoli, let's say 1 to 3 microns, that size is the perfect size for macrophages to take them up. And we have an army of alveolar macrophages in our alveolar air sacs that would just gobble these things up and destroy them. So that was the reason why it was thought, very difficult, if not impossible, to get controlled release. So particle physics supported the dogma, and that this was the deposition mechanism I was mentioning. If a particle was too small, also like really nano-sized, it would have high Brownian motion, and it could deposit in the airways before making it to the air sacs. And that was thought that the distance a particle would travel out of its stream towards the side walls to deposit was proportional to time and diameter to the 1/2. Sedimentation was another problem. As it's in the airways, it can sediment out and also impact, and that's proportional to diameter squared. And impaction is another way. Particles coming in and the airstream changes direction. If they have too much inertia, they just keep going straight and they impact and they come out of the airstream and they don't make it to the deep lung. And that's also related to how fast you're breathing, but also the diameter of the particle squared. So when I started working on this with Dave Edwards and Bob Langer, Dave had this idea that why can't we make it a lot larger if we go ahead at the same time and make it very porous so that it remains the same weight or even lighter than the small dense particle. And so in fact, it was known that the aerodynamic diameter, the way the particle should behave in the airstream, is equal to the true particle diameter, but multiplied by its density, divided by a reference density, which is just 1 out of water, to the 1/2, and then divided by gamma, which is a shape factor for a sphere that's equal to 1. So in other words, people had in their minds equated basically d arrow with the actual diameter, because most things have a density near 1. But what Dave was saying is if we can make this density, let's say 0.1 or even less, then in fact, the way it behaves in the airstream, a 10 micron particle might behave like a 1 micron particle, for example. And so this was a genius idea because large particles can aerosolize much more efficiently. And they'd be too large to be removed by macrophages. The question is, would they come out by all these inertial impaction mechanisms or et cetera, previous to getting to the deep lungs? But like I said, the way they behave, you can make them behave like a 1 micron particle, even though they're 10 microns was the idea. So I worked on this with them. We found that they could very efficiently aerosolize from the inhalers, give us a very efficient delivery into the deep lungs for a particle much larger than people thought was possible. And that when they got there, because they couldn't be removed, they could give us long-term delivery of drugs. And examples of this, I'm showing now also Bob Langer and Dave Edwards, who I worked with on this, and many others at MIT at the time. But we call these large porous particles, and we showed that they easily inhaled from low-tech inhalers. So we were getting over 60% respirable fractions, where the industry standard was something around 15% at that time. They are a very large size, so we got the long-term release. And for example, we showed in a paper in Science that we could deliver insulin to the blood of rats and control glucose for 96 hours from a single inhalation. And the longest that had ever been done was insulin could do that for 6 hours if it was encapsulated in liposomes and inhaled prior to that work. We Some of the work that I did was to find coatings. I used actually these lung surfactants to coat the particles, and that made them break up more easily and fly more readily. And that became a way that we actually make particles even without polymers these days. Many of the early particles were made with polymers. So based on a lot of this work and more, Bob and David started a company called Advanced Stimulation Research, or AIR. That was way back in 1998. It was a big success. Alkermes bought it for 115 million the next year, and Civitas bought Accorda, who had acquired some of the rights also later on. And Civitas actually developed the first approved product based directly on this technology. And you'll see the paper Science was 97. The first product was about 20 years later. But it's inhaled L-Dopa for Parkinson's disease, and it's helping a lot of people. So that was really my introduction to this kind of work. And when I got to Hopkins, I started being interested in many other things. And one was, instead of delivering drugs to the blood from the lungs, maybe I could deliver them locally in the lungs or other mucosal tissues. So there's a huge unmet need to deliver drugs more effectively just to mucosal tissues. And I'll tell you about those in a second. There was a big myth, though, and which I assumed was true at the beginning, that particles that stick to mucins, so these secreted mucus secretions, provide benefits compared to particles that do not stick. So with that in mind, let me just tell you a little bit about this. So mucus coats all entry points to the body that are not covered by skin, with the exception of your alveolar sacs that I told you about earlier. But that includes the surface of your eye, your respiratory tract, your nose, your sinuses, your trachea, your lung airways, your GI tract, all the way from your mouth to the other side, female reproductive tract, inner ear, etc. And the mucus is there really to protect the body from the constant assault by infectious and toxic agents. So it helps trap foreign materials and then remove it. We produce liters of mucus every day that is then cleared on the order of seconds to minutes to sometimes hours, usually recycled because it's a huge amount of energy to produce so much mucus, but we do that to keep our body kind of clean. And that makes it difficult also when you start to think about trying to deliver drugs using particles that oftentimes have sizes that are similar to pathogens. So if we could develop better ways to get sustained and controlled release at mucosal surfaces, there's a whole huge number of diseases, very important diseases that could be treated more effectively. And I'm just listing a few of them here. I'm not going to take the time to go into them, and there's many, many more. So the huge goal to try to do this. But just a little bit more about that mucus. I mentioned that it coats all your entry points to your body that aren't covered with skin. It's highly viscous and also elastic, which means solid-like. So it has kind of both a liquid and a solid type behavior. Just focus on the viscosity that most people understand best. Water has about a viscosity of about 100 centipoise. And glycerol is something maybe you've worked with a little bit or felt, and it has a viscosity about 1,000 times that of water. If you have normal human mucus, has a viscosity at low shear rates about 2,000 times that of water. And if you have a disease, for example, cystic fibrosis, your mucus could be 10, 100, or even a millionfold more viscous than water in your lung airways, for example. And stiff rubber is somewhat similar to CF mucus. It's also very sticky. Almost everything sticks to mucus. That's one of the main ways it does its trapping. But what we also noticed that some great work that was being published out of the Netherlands, actually, Nick Sanders and Joe Demister and all, we're doing some really beautiful SEMs, and they were showing that mucus is in fact a gel, and it's a porous network of mucin fibers and other proteins and lipids and so forth. But some of the pores they were suggesting were on the order of, maybe tens to even 100 or so nanometers. So it's a porous gel. And so long story short, I can't go into all of this, but my group became convinced that it made sense anyway that the dogma that you should stick to the mucus didn't make, to make things kind of last longer and spread out more evenly, didn't make a lot of sense to us, because that's what mucus is usually trying to do in order to trap particles and remove them quickly. And in fact, we know that certain things that, for example, certain pathogens like viruses, like the human papillomavirus, that are the most infectious pathogens, those pathogens all are able to not stick to mucus and rapidly penetrate the mucus secretions and get to the underlying tissue. So we were very focused on that to try to get longer lasting and also to be able to do gene delivery. So long story short, we figured out how to make particles not stick. And so we, on the left, I have a little video. These are just conventional polymer particles, these little bright dots. You can make them out of a variety of different polymers, or you can make them quantum dots, all kinds of things. And they don't move. You can see they're stuck in undiluted human mucus. But on the other hand, if you coat the particles with an extremely dense coating of a non-mucoadhesive polymer like polyethylene glycol, then the polymer's particles can move through mucus like they move through water. And that's true for all different types of mucus that we've tested, both in animals and in humans. And I've listed some types here. So we call these particles that move, we call them mucus penetrating particles, or MPP. So they're basically the exact opposite of what the dogma was saying that you wanted, which was a mucoadhesive particle. So to penetrate the mucus, now instead of the micro like inhalation, we really needed a nano size. So nano size and a very high peg density were required. And here's some of the very early work that I'm showing here. But just to give you an idea, if we look at the percentage of particles on the y-axis and how fast they're moving, which is a log 10 effective diffusivity coefficient. If they had, even if they had PEG, but it wasn't high enough density, then they were here in the white bars, orders of magnitude slower than if they had a very high density of the same PEG on the particle surface. If you had no PEG, it would be even slower. But this work was really showing that you had to get to extreme densities of PEG into the brush regime in order for these particles to move. So there's just some extra detail of that. So we had done this and we had made it with a variety of different polymers and even just pure drug coated, like made drugs into nano-sized particles, like crystals of drugs, and coated them with PEGs and things like that. And then the question is now, do they work better or the same or worse than the adhesive particles? And so this is a picture I like to show. This is actually a cross-section of a human GI tract from a cadaver. And you can see that when you don't have food in your GI tract, it's all folded up. So the lumen is in white here, and then you can see all the folds and so forth. And same thing here. Now this is, I believe this is a mouse. I was going to say a rat. So this is a mouse GI tract. You can see it looks very similar. In one case, we have mucoadhesive particles, and it's one of the colors, and the other one is the mucus penetrating particles, and it's a different color. And so sometimes when I'm in an audience that really knows about the mucoadhesive drug delivery systems, I ask them which one is which here. And so the answer is that the non-adhesive ones are the ones that spread out very well and go into all the different folds of your GI tract. And even when we get really blown up images, it appears that they can go into the crypts, so that they're kind of getting everywhere. Whereas the adhesive particles, they just get stuck in that secreted mucin, and they are then shed very rapidly and cleared down the GI tract. So it looks like there's less particles here, and that's true because they're just being cleared more rapidly. So we've shown this in a variety of tissues now. This is just a few. Same story, the MPP spread out well in the GI tract, the what we call conventional particles now. The mucoadhesive ones don't. Same thing with the lung airways, vagina. It can go and reach all the folds, whereas if it's a traditional particle, it doesn't. And we've also shown in a variety of papers that this improves drug delivery, and also it's required for effective gene delivery, obviously. the gene delivery, you have to get through the mucus to get to the underlying cells. So we've shown this in a variety of papers. Here's some of the early ones. We started focusing when in 2010, when I just moved from the engineering school down to the medical school to work at the Wilmer Eye Institute, on seeing if this could even be used to treat eye diseases. And that was very difficult because the eye, we blink every few seconds. And so we're squeegeeing off the mucus and kind of replacing that really quickly. So anything that would benefit for the eye would have to penetrate very rapidly. And that was a big challenge. So again, we just have these uncoated particles in the eye. They actually got aggregated up very rapidly into big blobs that wouldn't move. So those would be easily blinked away. And then again, the coated particles. And I'm calling out Sam Lai here as kind of the person. After Michelle Dawson started this work, Sam really kind of perfected the PEG coatings that allowed us to get mucus penetrating particles. So this work was actually intriguing to a company that we had started to look at developing the mucus penetrating particles for human uses, so callus pharmaceuticals. And these particles are now, we used to call them the MPP technology. It's now called the Amplify technology. So they made it sound cooler. And that's a technology so I'm going to show you now in a couple of FDA-approved products, Inveltis and Isuvas. So this is now callus data after we showed them that we could penetrate the eye mucus rapidly, and some preliminary data says that they lasted longer on the eye. They did a really nice study in rats, and in fact showed that the conventional particles were cleared very rapidly, whereas the mucus penetrating particles could last a much longer time on the ocular surface of a rat. And so Hongmin Chen, actually, she was a graduate student at the same time I was in the Langer lab. We had desks near each other. She became the chief scientific officer of callus pharmaceuticals. And she led up her group's work on this. And they were showing that this is showing corneal drug levels of a particular drug called loteprednol icabinate, which is the main drug in an approved drug called Lotemax. And so they started testing that on the eye and see which formulations could they give the best drug levels. And just taking this Lotemax suspension, which is actually a micronized type suspension, and comparing it to the nano-sized suspension that had mucus penetrating ability due to PEG polymers on the surface of the particles, they could find that they could get orders, much greater drug delivery into various eye tissues. This is just showing the cornea. And in fact, if you look at the area under the curve, it was about the 40x that I had said that we had seen in our labs previously. So based on this and a lot of other work, they did a series of clinical trials and led to the approvals of a couple of different drugs. One is called Inveltis. It was approved in 2018 for the treatment of ocular pain and inflammation. That's oftentimes dosed after people have had cataract surgery, which is the most common surgery in the world. So millions of people in the US every year, for example. It's the first and only post-surgical ocular steroid that can be given just twice a day. Before that, ocular steroids had to be given 4 times or more a day. And we know that most people won't take drugs that often. And so the effectiveness is not as good. So it's much more convenient and likely much more effective for these patients. They showed very low rates of adverse events. One that was incredibly exciting was that intraocular pressure results when they were taking this steroid were not different statistically than vehicle. Which is a really, great thing when you're delivering a steroid, because that's probably the biggest concern. And it's now indicated for all ocular surgical procedures, of which there was about 8 million in 2017. So, tens of thousands, if not hundreds of thousands or even millions of people have used this already. More recently, they were able to get Isuvas approved, which is a potential first-line treatment for dry eye disease and for flares specifically. And there's about 33 million dry eye sufferers in the US alone, and about 90% of them experience flares. And so Isuvas was approved, and it's also loteprednol tabinate, by the way, but at a much lower concentration. but approved in the US in October 2020. And based on 3 trials with thousands, combined, it was well over 1,000, I think over a couple thousand patients. So a huge number of patients go into these trials. Okay, let me go on to another unmet need that I've worked on, and that was the need for long-acting injectables for ocular disease. Reaching the inside of the eye, especially to get drugs to the back of the eye, is very difficult. And so people oftentimes are taking injections, for example, about once a month or every 2 months to treat blinding diseases like age-related macular degeneration. And what they really needed was a long lasting polymer type delivery system so they wouldn't have to get so many injections. However, when I started working on this, I was actually consulting with a major company, and they said that all polymer particles, even PLGA, which is polylactic glycolic acid, the material used in biodegradable sutures, even that causes inflammation when you inject it into the eye. So, they'd basically given up on that. So our goal became to figure out, is it true that all polymer particles would cause inflammation? And I didn't believe that, I believed that we should be able to find something that wouldn't cause inflammation. And so we started to work on it. The other thing that we needed to do was not only encapsulate and slowly release the drug from some sort of polymer particle that could be injected, but when that particle is injected, you can't just have them floating around in the eye, because they could disrupt vision. And you see it in your eye as something we call a floater. So we also needed to develop a way that once it's injected, the particles would aggregate. So it would go in through the needle like just a particle, so easy. But once it got in the eye, they would come together and form like a little depot in the eye. And then once they were there, slowly degrade and release the drug. So not a lot of detail here, but I had the idea that if we had those very dense PEG coatings on the particles, that they could prevent inflammation in the eye. And essentially that was based on what I had learned by talking to people and trying to find things to read that suggested to me that maybe the polymer particles that were being injected were adhering to the vitreous gel fibers, causing them to collapse and change and therefore causing a mechanical stress on various cells in the eye that would then signal the inflammatory cascade. So I said, if we make them non-adhesive to the vitreous gel fibers, similar to being non-adhesive to the mucus fibers, for example, then maybe they wouldn't cause inflammation. And in fact, so I asked Peter McDonald, who was the head of the Wilmer Eye Institute, if he could collaborate and test this in animal models. We tested it in bunnies. We basically had uncoated particles or lightly coated particles, and those caused quite a bit of inflammation, just like the dogma had said. But if we had them with a very dense coating of PEG, for example, so what we call surface treated, the inflammation that it was just the same as you get when you inject saline into the eye. So that was basically another story that involved very dense pegylation to eliminate, in this case, inflammation of injectables. So this was exciting. At the same time, at Hopkins, we were developing various delivery systems for various drugs using these heavily pegylated PLGA particles. One that became Greybug's, a company that I founded's lead drug, GB102, is sunitinib. And the main reason that we were interested in sunitinib was because it's a pan-VEGF inhibitor, because it blocks all the VEGF receptors. And so that could then prevent angiogenesis. And angiogenesis is responsible for multiple blinding diseases, including the most common blinding disease of the elderly, age-related macular degeneration. So that was one of the main reasons. It could also be useful in diabetic retinopathy and macular edema, which are hugely prevalent. But it also, beyond being anti-angiogenic, it also has neuroprotective properties because it inhibits the DLK pathway, and blocking DLK promotes retinal ganglion survival. And it could be therefore used in things like glaucoma and retinal therapies. Jay Fu, who I've worked with for over 20 years, amazing chemist and formulation scientist, developed delivery systems for sunitinib that could last a month all the way to well over a year. And this and a bunch of other preclinical work was published recently in Nature Communications in 2020, if you're interested. But Greybug moved forward in clinical trials and ended up focusing on a 1 milligram sunitinib dose, intravitreal injection to treat AMD. And these are actually some pictures, photos of patients' eyes that had received injections. This is actually one patient at different time points. And you can see the polymer depot here. It's colored here a little bit orange. It's in the eye, the particles are stuck together. It's out of the field of vision. So it's, well down in the bottom part of your eye. And at different time points, it's shrinking. Still at 6 months, it's still going and still has that orange color from the sunitinib. So it's still releasing drug. By the way, this has been injected now well over a couple hundred patients, and none have had any inflammation, to my knowledge, at all. So that worked. But also, we were able to deliver the sunitinib for so long that patients were not needing to come back to get additional shots for, in most cases, at least 6 months. Actually, more than half patients, more than 8 months. But in something like 88% for the 1 mg group, they could go at least 6 months without needing a second shot. Whereas prior to this, again, people were needing shots about every 6 to 7 weeks with the best drug, longest lasting drug at that time, Eylea. So this is just some details on that. They've actually done phase 1/2A and then a 2B study. This is just some results from the 1/2A. This is all the different doses accumulated. So when you accumulate all the doses, it was 88% at 3 months. But in the 1 mg dose, you didn't have people needing another injection. 88% of patients didn't need another injection even after 6 months. So this company actually we founded in 2011. This was a picture of my lab at the time. And here's the company when it went public on NASDAQ in 2020. But a lot of people wonder where this name Greybug came from. And I show this picture because at that time, my kids were quite young. Here's Grayson on the bottom left and Riley on the bottom right. And Riley, we always called Bug, and Grayson, we always called Gray. And so we named the company after them so they wouldn't mind that their dad was working so much. Okay, so another unmet need is improved drug delivery to and penetration within the brain. And this is a huge challenge, obviously, to get even any drugs into the brain because of the blood-brain barrier. Even small molecule drugs, very few of them cross, and when they do, oftentimes not to a great extent. Even more difficult to think about getting particles in a non-invasive way, and I'll get to that in a little bit, and maybe in the last myth. But if you're going to go ahead and infuse particles locally, there was a myth that particles had to be very small, like less than about 40 to 60 nanometers or so, in order to spread out, between the cells and spread out in the brain. And we thought that would be very important if we're going to really be able to reach larger portions of the brain, for example, to do gene therapies or even to deliver various drugs. So we, kind of skipping a lot here, that was a very strong dogma. But long story short, again, we showed that particles were non-adhesive. So again, with even denser PEG densities than we needed for mucus, in fact, was needed to be able to penetrate between cells in the brain. But when we got those extremely dense PEG coatings, we started seeing particles that were quite large being able to spread. And so we were doing at that time localized injections into mouse brains or rat brains of particles that were either coated or not coated with dense coatings of PEG. So unpegylated would be in red, shows up yellow because it's co-localizing with the green. So just along the needle track, if you didn't have any PEG, the particle, even 40 nanometers, don't spread. But if you have them coated, so coated that they can actually diffuse and move around. We call those brain penetrating particles. You can see right after injection, they're already starting to spread away from the needle tract. Same thing, even surprisingly, with a 100 nanometer particle. And so here's the needle tract again. The coated particles are moving away from the needle tract, so they're penetrating. And then we had a window in this, this is a live animal, and we had a cranial window, and we used confocal microscopy and tracked these particles. And even far away from the needle tract, you can see that they're not stuck to anything. They're able to move either by convection or by more slowly by diffusion. If you got to large enough particles, it didn't matter if they're coated or not, because there is a mesh size within the brain. And so that was about 200 nanometers. We saw no movement. So what was interesting here is that we showed that particles could be much larger than 40 to 60 nanometers. Even these were about 114 nanometers with the PEG coatings. So they could be much larger as long as they are non-adhesive. And that's important because it's very difficult to make the drug delivery systems with particles that are 60 nanometers or smaller. So this gives you a lot, as a drug delivery scientist, gives you a lot more leeway in making different types of delivery systems if you can make them as large as, say, 100 nanometers. So this was published in 2012. Elizabeth Nance was the lead author, and she's now a professor at the University of Washington. So we've gone on and done, a variety of different drug and gene delivery systems. This is just one, some early work. This is Panos Masterakis, who's now at UVA, but was a postdoc in the lab at the time, and he was developing gene delivery systems. So in this case, instead of using PLGA, we're using cationic polymers to condense genes down into in most cases, we were making sub 50 nanometer gene carrier particles. And they were either densely coated with PEG or lightly coated with PEG or not coated with PEG. Here, we're actually looking at the particles being labeled. The ones that are showing up yellow have a low density of PEG, but a PEG density on gene carriers that was about the highest at that time. And then our systems that had a much higher PEG density are shown in red. And you can see the infusion was here. And you can see that mostly along the needle tract, a little spreading because of the PEG of the conventional PEG, but much better spreading if you had the very dense PEG. And here's just a quantification of that volume of distribution. I should also mention, in addition to Panos, Jung-Soo Sak was key in all of this work as a graduate student. And he had developed methods to make very, very small gene carriers that had a lot of PEG. And that was very difficult at the time because as you tried to put on more PEG, we were typically decreasing the charge density of the polymers. And so the particles would get larger. But he showed by making blends of the polycations with PEG and without PEG that you could make very small, very densely PEGylated particles. And that was a key innovation as well. So we've shown in a variety of studies that we can get transfection, for example, from a single infusion throughout the entire rat striatum. So here's if we have no PEG. I'm going to focus down here. This is a 3D rendering of many slices through the brain. So no PEG, some PEG, and then dense PEG. You can see that you get, this is now looking at where is the gene being expressed. So where you see green is actually a gene product being made. And you can see that it's basically throughout the entire rat striatum. The question is, how much larger can we go? How much larger brain can we still get transfection throughout? And one of the main reasons I'm interested in that is that, eventually I would like to be able to think that we could deliver genes to almost the entire brain, but have them regulated by, for example, cancer-specific promoters, so that they would only be turned on in cancer cells, and that this could be used for something like brain cancer. We're also quite interested in using these in Parkinson's, and that's going to be at the end of my last unmet need, which is non-invasive and site-specific delivery into the brain. This is a big challenge on its own, but especially if you want to deliver something as big as a nanoparticle into the brain from the blood, for example, that was thought to be very difficult, if not impossible, to do that effectively. So the myth is that drug-loaded nanoparticles cannot be safely delivered to specific sites in the brain non-invasively. When I was a young professor, this is about in the year 2000, I believe, I met Rich Price, who was also a young, enterprising professor at the University of Virginia. And he was working on using focused ultrasound to transiently permeate different vasculatures. At that time, they weren't doing brain, but we started a collaboration. It was a good collaboration. And then some years later, he called me and said, that he thinks that he could do this in the brain now, that they had developed systems that you can use focused ultrasound, that they can do it from outside the brain, obviously, and they had figured out how to do it, correct for the fact that you have a skull, different thicknesses and so forth, and still be able to get sub-millimeter precision with where they would be hitting with the high intensity focused ultrasound. So a focused ultrasound is basically causes a pressure wave. And so, high pressure, low pressure, high pressure, low pressure locally in a tissue. And again, they can focus it to sub-millimeter resolution. And so if you inject into the blood particles that are contrast agent particles, which are called microbubbles, they're just albumin, shells basically that are filled with air. And those are used for ultrasound contrast agents all the time in people. But if you put those into the blood and then choose where you want to transiently permeate the vasculature and hit it with precise types of focused ultrasound, what happens is those pressure waves going through those microbubbles cause the microbubbles to squeeze down during the high pressure and then expand during the low pressure. And so they basically, in the area of focus, the microbubbles coming through are doing this. They're expanding and contracting very rapidly. And that process had been shown over many years by Rich Price and others in the field to give you a transient opening of vasculature. And the field was developing to do this also in the brain. So we started working with Rich, and we started asking, could we take these very heavily pegylated particles that were gene carriers, and since they're heavily pegylated, they should circulate a reasonable amount of time in the blood, but can we get them to go from the blood into the brain and transfect tissues locally in the brain? And so this was just some early work where we were trying to figure out what kind of dose we would need. And we were using in vivo fluorescence measurements to look at a gene that was producing a fluorescent molecule. I can't remember which one now, but suffice it to say that if you see a enhancement here, it's because that you have a gene product being made. And so we could see it even when we delivered just 50 micrograms total to the whole body of DNA in our particles. We got bigger with 100, more with 200, and even more with 350 micrograms of DNA delivered. We also only see significant transfection where we use the focused ultrasound. And of course, some ways away from that as the particles start to spread and diffuse. So this was actually, if you take the brains out, the signal got even stronger. And you could see it here in these same rats, that you're getting quite robust expression with just opening the blood-brain barrier in one area. We've gone on and done a lot of different work. I'm summarizing very rapidly, but this was an interesting paper in 2017, and where we put in a gene called GDNF, which is a neurotrophic factor that can help potentially to regenerate dopamine levels in the brain if we could deliver it effectively. That was the goal. And so this is actually very complicated, but what you're looking at here is basically looking at the GDNF expression. So this is the gene that we're delivering. So it should only be high if the gene is really effectively delivered into the brain. And we're looking in the side where we deliver the gene is black, and the other side of the brain is gray. It's kind of a control in a sense. But you can see after 2 weeks, which is when I believe we do the dosing, very low levels. And then after 6 weeks, you can see very high levels, but only when we deliver deliver these particles using focus ultrasound, the GDNF in the brain penetrating particles that are administered systemically, and obviously not when we have a control plasmid. So we could get very high levels, for example, at week 6, and that is either about the same, if not even higher, at a week 12. So very long-term expression, and that has to do somewhat also with the design of the plasmid, making sure that once it's delivered, it does not get silenced. So in that work, we also then, it was exciting that we were delivering so much GDNF. We started looking at whether or not the neuron density could be restored in animals that we had poisoned the neurons using this molecule 6-OHDA. But what we found was that the darker staining is actually due to the neurons. And so what we found was that it was getting a lot darker and better if you delivered the gene using focus ultrasound, and didn't work with all the controls. And also in the substantia nigra pars compacta, which is an important region in Parkinson's, we could see that we could, in fact, regenerate these neurons. So this is just a summary of that. Again, a very complicated slide, but in various ways looking at, looking to see if we could regenerate the neurons. We found that at week 6 and week 12, that we were actually getting quite good staining for the neurons, etc. So we were actually, restoring the density of these motor neurons. But that's good to see them. Were they, the question is then, were they functional? Were they, able to secrete dopamine? And in fact, we found that they were. And so the contralateral is a side of the brain where, has normal levels of dopamine, because we didn't poison the neurons on that side. So the gray bars is what you want at 6 weeks and 12 weeks. And here's week 2, right? So week 2, before we started treatment, our animals were down here with dopamine, where they should, in that side of the brain, where they should have been here, like the other side of the brain. But only in the treatment group that had the focus ultrasound and the GDNF, we started to get levels by week 6 that were much higher than background, and actually starting to approach normal. And by week 12, actually quite, maybe not quite, but getting quite close to normal in dopamine levels as well. So Rich did, in his lab, did some functional tests as well. And one is, in these animals, you could, since you're just poisoning one side of the brain, if you then hit them with a stimulator, they will start to rotate, and they'll basically walk around in a circle, in a circle, in a circle for some time. And the more that they walk around in the circle, the higher the level of neuron death that they have in that side of the brain. So you want low levels of this rotation for better treatment. And so at week 2, these animals all were rotating quite a lot, like about 7 times per minute or so. And then we start the treatment. So this is the gene therapy / focus ultrasound treatment. And in all of the control groups, so animals that just had the poison, no gene therapy, animals that had the poison and focus ultrasound, but a sham gene therapy, and animals that had the poison plus the GDNF gene therapy, but no focus ultrasound to get it into the brain, they all continuously got worse. And by about 4 to 6 weeks, it leveled off somewhat, somewhere between 10 and 15 rotations per minute. And we were hoping to just be able to keep at least this steady in animals that got treated. But what was remarkable that that kind of went along with all that neuron density staining and dopamine production was we saw an improvement, statistically significant improvement from baseline in the animals that received the gene therapy. And another way that you could test this is when you poison just one side of the brain, and then you put a clear jar over a rat. The rat will try to feel its way out of the jar, and it'll start touching the jar. And if it has a really severely, low motor density, low motor neuron density in one side of the brain, it will touch the jar a lot less with the opposite hand, so that you get a bias towards one paw versus the other. And so normal would be that it would touch equally, 50% with the left hand and 50% with the right paw, I guess you would say. So in doing this, here again is there's a bias. It's touching about 3 to 1 with one of its paws versus the other in all of the groups prior to treatment. And then all of the groups don't get any better after that. They actually didn't get any worse. But in this case, if you treated, the 4-paw bias went back to, essentially 50-50 or normal. So this is something that we're quite excited about and hoping to develop into a treatment in the future. Some conclusions related to the brain work: that the mesh size is much larger than expected, so that should help drug delivery scientists. That the extreme PEG densities allow it to spread once it's in the brain, so it can reach more cells and deliver things like genes more effectively to more of the cells in the brain. And that the MR-guided focus ultrasound can allow targeted and safe DNA delivery into the brain. By the way, I should say that focus ultrasound in the brain is not just in animals now. Hundreds of patients have undergone focus ultrasound in their brains safely, human patients now. So to my knowledge, nobody's delivered a nanoparticle yet into the brain. We were the first to do it in animals with Rich. Well, Rich, I should say, was the first to do it with our particles in the brains of animals. But hopefully somebody soon, if not us, will be doing this with nanoparticles in the brain of humans. So I just have a few take-home messages for my talk today. For the young folks out there especially, is at least in my life, my career, I've found identifying unmet needs that have huge potential impact, and really kind of thinking through which are the ones that require the most attention, that you feel like you can do something about, and prioritize those. That's an important first step. And then another one is to question some of the dogmas out there. I think we too easily accept what we may read in a paper that was said oftentimes without really strong scientific evidence, and then that sometimes gets propagated through the literature, and that can be very stifling to innovation. So always make sure that you are convinced that a dogma is correct. Don't just accept it based on the words of even someone famous. Test that dogma and systematically solve the problems along the way. And what helps you really know if even if a dogma could be right or wrong is really diving in and understanding the biology and the physiology. I mean, I was a trained chemical engineer, and I could make lots of whiz bang, nano things and so forth. But the big difference in my career was when I really realized the power of understanding the biology and the physiology, in addition to being able to make really cool things. And then I would also say, don't favor the pet approach. Oftentimes we have even our own pet reasons or things that we think work best. And we shouldn't favor that. We should be willing to change and develop new systems, or even, realize somebody else is doing it better, and what can I do to even help make that better? And that can oftentimes get us unstuck and allow us to innovate. And if you do these things, I'm hopeful that you will be able to innovate and create breakthrough products in the future. Literally, as I went through this has been my career over the last, since 25 years or so. I realized there was just hundreds of people to acknowledge. I tried to therefore acknowledge some of the really important key people along the way. Of course, I'm not able to mention all the countless amazing graduate students and postdocs that have come through my lab that have helped. all of our collaborators, which has been well over 100 PIs, and then all the people in their labs over the years. Just so grateful for all of them. I like to say I didn't, except for when I was at MIT, I didn't get in the lab and do any of this. It was all done by this amazing group of students and postdocs and collaborators. And I've been privileged to be a part of that. So thank you very much, and happy to take some questions or start the panel.
Well, thank you so much for that amazing talk. We really appreciate it. And now we'll have a time for discussion, but let me first bring out our panel. So first we have your colleague, Dr. Hangang Kui, who's a professor at Johns Hopkins with appointments in the Department of Materials Science Engineering, Department of Oncology, and the Sidney Kimmel Comprehensive Cancer Center. He's a fellow of the American Institute of Medical and Biological Engineering and associate editor of Journal of Controlled Release. He's won a number of awards, including a National Science Foundation Career Award and the 3M Non-Tenured Faculty Award. And next up, we have our amazing host, Professor Alice Zhang of Peking University. And we have UCLA alumnus, Dr. Jianting Ding, who is a professor at the School of Biomedical Engineering and deputy director of the Institute for Personalized Medicine at Shanghai Jiao Tong University. He's in the Chinese Top Elite Researcher Program, the Qixi Foundation Elite Young Investigator Program, and was named a Southern California Scientists and Engineering Association Elite Young Researcher. His research interests focus on developing personalized therapy and precision medicine, including developing biosensors for early detection of cancer, infectious disease, metabolic diseases, age-related diseases, cardiovascular diseases based on cellular, electrochemical, impedance, mechanical, and photonic signaling, as well as developing personalized treatments. He won the ICANX Young Scientist Award last year. And then our ex-challenger today is Dr. Woojin Sun, who's an assistant professor in the Department of Biological Systems Engineering at Virginia Tech. He also came through UCLA as a postdoc and worked with one of our alumni and former colleagues for his PhD as well. His research interests are at the interface between cell engineering and biomaterials engineering. He's interested in applying synthetic biology tools to engineer cells for therapeutic applications, and also in developing drug delivery systems such as nanoformulations, wearable devices, and injectable drug releasing materials. He aims to integrate synthetic biology, drug delivery, and tissue engineering techniques to design precision medicine. So let's bring everybody out. And as is traditional, we'll start with our ex-challenger, Woojin.
Thank you, Paul. Thank you, Justin. That's a really inspiring talk. I'm always excited about the idea of translating what we do in the lab into the clinic. But as a beginner who just started my lab, I find it not easy to decide what disease should I target or what kind of delivery system should I stick with. So what would be your advice for beginners?
Yeah, thanks, Woojin. That's a great question. I don't know that there's one right answer, but at least for me, I've always been interested in cancer, because I feel like that's such an unmet need. And so I have done a lot of work actually in the cancer area, largely because it's such an unmet need. We all know that, right? We need better, the drugs we use for cancer are somewhat barbaric in many cases, chemo poisons and things like that. And we know that we could, potentially improve that if we could just deliver a lot more of the poison to the right cells and a lot less to the rest of our bodies. But I also encourage people to really think a lot about other diseases, because there's so many, and so many that have really, that are huge problems and have very poor solutions. And in my case, what I'm good at is, designing delivery systems that can get to difficult to reach places oftentimes, and then provide drugs, in a sustained fashion, or maybe even enter cells to deliver things like gene therapies. So out of those really important sets of diseases and different organs that I learned about by talking to collaborators and inviting people to speak and, having conversations with clinicians and scientists and so forth. Out of those things, I start to think, well, out of all these huge unmet needs, what are ones that I think that I can most readily, with my skill set, have a, impact? And so, and sometimes I don't even know. So I have to share what I can do, what I know with various experts. And they can oftentimes say, wow, could you do this? And then so for example, in the collaboration with, that led to GB102, I had learned that people could not use polymer particles in the eye, they thought, because they're inflammatory. Well, I thought I can do something about that. And I could see all the potential advantages of, with all the different drugs that could be delivered. But really the idea to use sunitinib at first came from a colleague at Wilmer named Don Zach, who had identified it in a screen of 10,000 plus compounds as one that was strongly neuroprotective. And neuroprotection, the way we treat glaucoma right now is by lowering ocular pressure. But a lot of people with glaucoma don't have raised ocular pressure. So you need something that could also just protect the eye even when you don't have high pressure. So it was like, kind of really exciting to think about choosing that. And then therefore that leads you to start with glaucoma. And then of course, the more I learned about the molecule, I learned that it was strongly anti-angiogenic properties, VEGF inhibitor. So I asked Peter Campachero, do you think a long-lasting anti-VEGF would be, something that would be interesting? And he was one of the lead retinal doctors at Wilmer, and he said, absolutely, 100%, let's test it. So I think it's a little bit of a long answer, but there's, you can look at almost any part of the body and there are unmet needs. So then I would say one of the most important things, if, a lot of the folks here are probably more on like the chemistry or engineering, nanotechnology side. One of the most important things is to really start talking to clinician scientists about what you can do and learn a little bit about what they do and get some ideas of how your systems might be helpful for them, but then also listen to them and work in partnership. Collaborate. Don't worry too much about if you're getting enough credit or anything. Even as a young person, sometimes that's very hard because we're trying to prove ourselves. But if you really want to have a major impact, if you can get the best people to work with you, you'll have more impact, and then you will become famous, or whatever, right? So don't worry about that. Just bring more value than anybody else, and be selfless about it, and see what you can really achieve in your life. Is what I would say, basically.
Yeah, thank you, Justin. You just mentioned the importance of collaboration. I believe you have many wonderful collaborators. So what's your strategy in identifying the collaborators? And also, I'm also curious to know if you have any experience about failed collaborations.
Failed. Okay, yeah, sure. I think, I've collaborated with well over 100 different, faculty members and also industry. I don't mean to not say industry, but, I'd say the vast majority of those were very productive and good. And some, just didn't work out perfectly, like, whatever we were doing didn't work and it fizzled out. Very few, there would be, there was just a misalignment, I think, on what was either important or goals and so forth. Or I'd say that probably the most, one thing that can happen that can hurt a collaboration is if you don't really, if either side doesn't really appreciate what the other person is bringing to the table, so, appreciating your collaborators is very important, I think. So how do I find them though? I mean, that's a good question. I think that, I like to go to a lot of talks. I like to, and I like to find people who are, probably the first thing I do is if I have an idea that could be used in a certain area, I want to know who are the biggest experts locally at Hopkins. And from there, who else should I talk to? I try to talk to experts who can point me to other experts. Now, I will ask people, what does that person like to work with? Because it is important. And then I'll try to get to know them a little bit. And I think collaborating with people who you can become who you like is really, really helpful. I have also collaborated with people that I probably wouldn't hang out with a lot on the weekends or something, but were some of my greatest collaborators too, so be open to that. I think I look for excellence. I look for people who are kind, gracious, grateful, because that matches me and that gets me excited. And I think that they kind of would lead their groups in a similar way that I would lead mine. I don't want to make it too complicated. I think, there's just a good feeling that you get if you're working with somebody that's going to be a long-term good collaboration. You just get a good feeling every time you talk to them, about science or whatever. So.
Thank you. Thank you, Justin.
Maybe the rest of the panel could chime in on that one because that's a good one. Alice.
Alice, yes.
Okay. Yes. Excellent talk, Justin. Congratulations first. Actually, I want to ask. I already take a question from my colleagues. Yeah, because this morning I have a meeting with someone in the hospital. The PKU has a very good hospital too. So I told you will deliver a talk, on this stage. I talked some story about you, like, medicines developing and all these company stuff. So the one doctor said to me, you must ask this question to Professor Justin for me. Say, yeah, how is the Johns Hopkins University, does they have any special strategy to enhance the collaboration between the people in the hospital and for the research? Does Johns Hopkins have some special strategy or policy?
Yeah, boy, I'm probably not the, I guess I have a lot of experience, so I can answer that somewhat, but there's probably people who can answer even better. But for sure, there's a lot of different things that Hopkins does to enhance the collaborations between faculty members. And there's like amazing institutes, for example, like the Institute for Nanobiotechnology that Honggang and I are part of, that span the entire university, that where nanotechnology and medicine's just one aspect of it, but where they merge and there's lots of people involved and working together through that. There's grant opportunities internally. There's our biomedical engineering department was originally housed in the School of Medicine and now has footprints in both the, what we call the Homewood campus, which is arts and sciences and engineering, and also the School of Medicine, which those are also located quite close together. There's a shuttle bus that runs between those two every 15 minutes to allow people to go, between to collaborate. So they do, I think, just a few things. Honggang maybe can add some more, but yeah, I think the university itself believes it's quite important to have these synergies.
Yeah, so Alice, if you can add a little bit more, I think Justin is too senior to know this in the program. The provost office, they have this discovery grant. In her grant, you have to apply from 2 different divisions. One school medicine, one engineering, one art science. And also, a few years ago, we have this blended days, for the junior faculty, between, engineering school and also School of Public Health. It's like 5 minutes. There's like basically a round table. You meet with someone from School of Public Health, you talk 5 minutes, you move to the next one. So they do put out effort to promote cross institutional, collaboration at Hopkins.
Okay, that's great. Actually, that's how PKU was promoting, the collaboration in this way. I will ask them to follow up. Okay, thank you very much.
Alice, do you have another question while we're with you?
If okay, I can ask one more question.
Please, yeah.
To Justin. Actually, this is a question, and 2 days ago, International Women's Day, March 8th, it asked on the stage for how the female scientists balance the life and the work. So they say this is a question often asked to the female scientists and never asked to the men. So yeah, here, yeah, it will possibly, I will ask you, instead of you putting your boys and the girls' name into your startup company, so anything you do to balance the work and the family? How to balance this?
Honestly, I was not good at that for many years. I think, I've just worked way too much in some ways, and I've realized, that I need to get better at saying no sometimes, and I need to prioritize, my own, kind of life outside of just, my work. I mean, the thing is, I love my work so much, so it's like and it's like another family for me, and I'm married to that too. but yeah, how do I manage the work life? I think I just, I'm not good at it, but I try to, set expectations that I'm not always going to respond to, right away on everything, or I can't even respond to everything. In fact, people, I think, pretty much know now that it's hit or miss to try to get me by email. I mean, I used to always just make sure my email box was clean, but now I'm sure that Paul and the others in LSU are aware. I mean, getting hundreds and hundreds of emails every single day, it's impossible. And so people, I don't give my phone number to very many people. And so I tell people, if you really need me, text me, and you can say that I sent you an email, please look for it, or whatever. But that's one way I've kind of helped balance my life. The other is I just make more time. I schedule in more time to do things with family, and to go on walks and to go to the gym, and it's things like that. I schedule time for myself, and I take time just to also just let my brain totally veg out and relax, because you got to clear, for me at least, I have to clear the clutter to be able to really think. If I'm just always doing the next task and the next task and checking off a huge list of things to do, my day is over. I'm depleted of energy completely. I did probably nothing creative, probably nothing that's really going to make a difference.
Okay,
and so,
thank you for your answer. Yeah, actually, that everyone knows that it's not only a problem for the female scientists, also a problem for the other scientists. Yeah, thank you very much. And the list, I love the Greybug. Yeah, the idea is great.
And let's go to
Can I chime in and ask a question?
Of course.
Yeah, it's very great talk indeed. I did learn a lot. And the question I think is I do see that you have a lot of companies. Okay, how do you balance between your research laboratory work versus your company? And that's the first question. The second question is that in your view, do you think professors shall go to the company and operate the company by themselves, or it's better to look for a professional CEO who help you to take over the company and do the routine stuff? What do you think? Thanks.
Yeah. So the first question is, how do you balance it? It depends. A lot of times when we start these companies, when they're very young, there's a lot of interaction. And for example, in a company that we started not too long ago, we're just about to complete a sponsored research agreement with the Center for Nanomedicine at Hopkins, so that we can continue to do certain kinds of work together in a seamless fashion. And Johns Hopkins, you have to always report this, and you have to be very clear to all your trainees and stuff what's going on. But Hopkins has been very good about working with companies like that and in monitoring conflicts and so forth along the way. Usually the companies, they'll take your technology and they'll start developing it for maybe at most a few products. And oftentimes the companies are very happy if you're trying to show that the technology could work for other products or more difficult things. And, so that's not a problem. We're not ever prevented from publishing. Like any of our students who work on projects related to the technology that have been licensed to a company, they're never prevented from publishing or anything like that. So, how do you balance it? It's just, you have to be careful about it. You have to be, upfront and disclose everything about it. And then, ideally you communicate a lot, over communicate, and make sure that people are on board as much as possible, and that if students have any questions that they can come to you, or if they feel more comfortable, they can go to a colleague or to any, to the head of the, whatever, right? Make sure that they feel very comfortable. Did I answer both, or is there a second one there?
The second question is, do you operate your company by yourselves, or you prefer to look for someone else? And how do you find this?
I'll tell you, I don't think it's a good idea if it's your first company to try to operate it by yourself. That's been, I was, I don't know if I can give myself a pat on the back or whatever, but I feel like I was smart enough in my first company to ask my former mentor, Bob Langer, to be a co-founder with me, because he had done it so many times before, and just so many things you don't know. And so we did that, and right away we went out and got venture capitalists, and they put somebody in as the CEO, and that was great. Other companies, maybe the technology is a little newer, and you're still thinking that it's going to be hard to raise a lot of venture capital for a while. Maybe I can write some SBIRs and things. Then Hopkins allows us for a couple of years to serve in an operating capacity, as long as the amount of time is limited. And so I've done that in the past. For example, with Greybug, I was the CEO for the first couple of years, and I'm serving as the CEO right now of Novus. But the goal usually there is, to basically find a great person to take over. So, your happiest day is when you fire yourself as CEO and you hire somebody who's a lot better.
Right, exactly. Thanks.
Very good. And let's move on to Honggang, Richard.
Good. So just I want to ask you a top question about particle size, right? In nanomedicine, the size matters. The size is important. You show that for inhalable delivery, 1 to 3 micrometers. Then for your mucus penetrating particles, right? So you have a nice range. The most material scientists or chemists, they try to make a particle same size. But in reality, we cannot make a particle same size as well. In the field of polymeric material, people know that polymers of different molecular weight could contribute positively to a material's property. You can make materials with different molecular weight. They can have toughness and strength, right? Then my question for the panel also as well. So when we talk about nanoparticle, right? Do we really want to make a particle of same size, right? Or particle dispersity to some extent can really benefit, the drug delivery. If you think about your brain tumor work, you inject them particle of different size, they could cover a larger area because of the size could distribute different range. How do you think about that? The particle size, the impact in drug delivery research overall.
Yeah, no, I think it's a very good point. And particle size distribution, for example, just if we think about inhalation, I always thought, although I don't know, that I've really acted much on it, but you might want to deliver drugs, throughout the entire airways, and in which case you might want a mixture of some larger that are going to come out a little earlier, some medium size to come out in the middle somewhere, and then some very small size, at least aerodynamically sized, so mass and basically mass in the end of the particle, to make them come out in different regions. Also, maybe different sizes might, in the case of the inhalation, it's coming out of a dry powder inhaler. What is it better to have all the same size particles, or to have different size particles to, for things to break up more efficiently and so forth? Things to think about there. You could think in certain cases, very small particles might dissolve a little faster and give you like an initial kind of loading of drug that could be very helpful. And slightly larger particles take longer and could provide the more sustained effects thereafter. So, just thinking off the top of my head, I could think of lots of reasons why, you might want, in some cases, very precise, sized, very small, tight size distribution. In other cases, you might want it to be more broader. I don't know if others have the same kind of experience or thoughts.
And Honggang, did you have another question, or may I bring a question from our audience forward?
I think I have another question, but I think we should give opportunity to the audience.
Go ahead. You can go ahead. Yeah, we have. I'll follow up after that.
Okay. So I want to bring back to translation. Justin, you are incredibly successful in translating the work from your lab, to commercially available product to companies you have funded, right? Greybug and Callus Pharmaceutics are a publicly traded company at NASDAQ, right? At what point of your career you start consider seriously, this path to commercialize, your research, and what do you think, the key factor that make you successful in doing so?
Yeah, well, I was, I did my PhD with Bob Langer, who started dozens of companies, and so I was kind of steeped in that environment that this was possible, and frankly, it kind of excited me, that I could work with all these young people on tons of different ideas, and the best ones we could either license them to companies, or if we needed to start a company and develop products out of them that could help people. So I always liked that idea, but when I started my faculty career, I thought, okay, I'll probably start a company at some point, and about 5 years in, actually some venture capitalists approached me about one of the papers we published, and really wanted to start a company on it, and even offered what I now know is pretty good terms. I didn't know then if it was good terms, but in hindsight, it was really good terms. And but I didn't do it at that time because they didn't see it, but I could see that technology was so early that we needed to do more before I was convinced that we were going to be able to make a good product out of it. And I didn't want my first company to be a flop, because I felt like fair or not, I mean, if my first company is a success, then I'll probably be able to start more, and if it wasn't, then you might have a more hard time. So in fact, I didn't do it. I was also very busy trying to get promoted and all that stuff. I was just 5 years in. So then about the time I got was up for promotion to full professor, another venture capital group called me about a different paper, and that actually was a paper by Sam Lai and PNAS for the mucus penetrating particles. And at that time, that technology was also quite new, but so I said, I said no at first, but I called them back about a year later after we had, kind of expanded that technology, and said, that I was ready to go if they were. And so that's how that got going, actually. And at that time, I'd just been promoted. I had never taken a sabbatical, so I took a year-long sabbatical, and I worked on it. And I did my sabbatical at Hopkins, so I still worked on Hopkins too, but at least I had more time. And it took all that amount of time, especially then, because, it was new to me doing all of that. So it's a major undertaking, but after you've done it, once or twice, then it's a little bit easier. It's still hard. I would say my biggest advice there is the first one you do, make sure you do with somebody else, ideally as a co-founder with you who's done it before successfully, who can help you, with all kinds of, they'll add way more value than you'll ever give them credit for, essentially. And they'll help you be successful.
Thank you. And we have a question from the audience, which is back over to science for a moment, and then we'll go back to life with mine. And that is, could you comment on the future of exosomes in drug delivery?
Yeah, certainly there's a lot of potential with exosomes, and already, for example, in diagnosis and early diagnosis and so forth of different things is in cancer and other places is huge. Their potential, what we can learn from them and use in drug delivery, the potential is enormous. I have followed that a little bit, but I'm not an expert really in exosomes, so I don't want to, say that I have all the answers to that question. But from what I've seen, it's an exciting area.
Mm-hmm. And then we have so many trainees listening today. I wonder if you might go back to when you were an undergraduate and a graduate student. And I remember when you spoke to my class a few months ago, you really, put yourself into your studies and trajectory. And if you might share a little bit of what you came into university with and how you rose to such success through your efforts then.
Okay. Wow. Yeah. Let's see. So how I came into it, came into the university. I was I think I was the first person in my family to get a 4-year college degree. So, in fact, at first I wanted to just go and work for my uncle, and my mom said, no, you can, and I probably would have been like digging ditches and things like that for a long time. My mom said, no, you're going to go to college first, and then after that you can decide, what you want to do. That, kind of came into it with that. I liked chemistry and math, so I started in chemical engineering. I started to like medicine. I thought I would go into medicine, but I had a class with a professor there, someone Paul, Hal Mombuket, that started talking about biotechnology. And I started thinking, wow, if I did something in the area of biotechnology, whereas if I was a physician, I might be able to interact with and impact thousands of people's lives. But if I do something really important in biotechnology, I might be able to impact millions of people's lives. And so that was, I applied to graduate schools like last minute to do that. And just so fortunate to have gotten into MIT and then met Bob Langer. And boy, did that just expand my whole, vision of what was possible. Is there another aspect that you were kind of wanting me to hit on, Paul?
Well, that was really, yeah. I mean, of course, there's much more, and we've had much longer conversations about that. But I do want people to hear that, our paths aren't smooth, and through your efforts and looking around the world, you were able to make yourself a success, and those around you successes by those by what you've done.
Yeah, one thing that
you told Alice, I think what you've done now with your with your life, it really shows how much you've been able to do both in your science and engineering and business on the one hand, but also as a person.
Oh, thanks. Thanks so much. Yeah, I mean, I think that what was interesting about me is I did, no one in my family had really done 4-year college, and people thought I was, even when I went to graduate school, people thought, what, are you ever going to get a job? And I just remember, being so motivated to do something to help a lot of people because, my grandmother died when I was 10 of cancer, and she was too young. And, I found my purpose in life, basically, that I wanted to do something. And, but, when I got to UCLA, I was not nearly as prepared as everybody else. I mean, I felt like, I was probably one of the, average at best, if not a dumber student in the chemical engineering program. So at that point, I just decided I would work harder than everybody else, and I just, because I felt like I had to make up for, my lack of intelligence somehow. And so I just worked harder. I worked as hard as I could. When I got to MIT, I felt the same way. I felt like, I don't know how I got in here. I don't really deserve to be here. I better work hard, and, so, I guess what I'd like to say is, a lot of people say, I'm a white male from middle class background. I've had all these privileges in my life. But even if someone like me can feel like, an imposter and not belonging, then it helps me understand others feeling that way. And what I would say is just, there's going to be times in your life where you're going to feel that way, that you're not the smartest or, whatever. And just realize that if you work hard and that if you take the attitude towards just, bringing whatever your best is and adding as much value as you can, and not being afraid to work with people who you feel are smarter than you. Like I feel like most of my collaborators are much smarter than I am, including Hongong. Chui is much smarter. Not being afraid to do that, not being afraid that you won't shine somehow, that is, helped me a tremendous amount. And, I end up getting way more credit than I deserve in the end, which is, ironic.
I will say, having spoken to my colleagues here, their impression of you is very different than what you felt yourself about your intelligence.
Exactly.
Stature. So
I feel you're much
think,
smarter than me.
Yeah, and we really appreciate, your not only your sharing that, but your really lessons for other people on humility, and also, I think for the many engineers and scientists out there who want to contribute to medicine, the idea of listening to what the problems are, and it takes that. I think it takes that kind of humility to do that, and that to me has been one of the keys to your success, and something we try and emulate here. So
thank you very much, Paul. It's very kind.
Yeah, well, I think with that note, let me thank our panel, and then I will hope to see you in person in the next few days. But normally, if we were all together, I could walk across the stage and hand you a plaque and thank you for giving this wonderful talk and showing us your amazing accomplishments, and also sharing so much about your life and strategy and science, engineering, and medicine. So thank you for this, and we'll look forward to seeing you both in person, and then also hopefully soon at UCLA. We have coming up in next month this ICANX Summit with luminaries including Nobel laureates, Gang Chen, David Lay, and others. So please stay tuned for that. Abstract submission deadline is, I think, next Tuesday. Oh, yes, here, March 15th. So beware the Ides of March. And on the next slide, you can see that next week we'll have Professor Sung-Woo Kim and Professor Alice Shang will be hosting. And so thank you all again from Los Angeles, from Beijing, from Johns Hopkins, from Shanghai, and around the world. And good morning, afternoon, and evening to you all. We'll see you next week. Thank you.